Welcome to MHC Molecules Series. This is episode 5. I am Dr. Kalpna Rakholiya and today we are going to discuss about exogenous pathway. It means MHC Class II Antigen processing and presentation pathway. In previous lecture, we already discussed about endogenous pathway. In exogenous pathway, you have to remember three things. It is the most important. Here, a pathogen or antigen are the extracellular. It may be bacteria, fungi, or toxin. Second thing you have to remember MSC class 2 molecule only present into the professional antigen presenting cell, that are dendritic cell, macrophage and B cell. Third thing you have to remember MSC class 2 only represent peptide to the T helper cell having a CD4 positive receptor. So let's start about the exogenous pathway. So here this is the professional antigen presenting cell. It may be dendritic cell, macrophage or B cell that will take up the extracellular pathogen via endocytosis process or phagocytosis process, and inside the cell with the membrane it form the endosome, or we also called a phagosome. This endosome and endosome or phagosome which are present into the cytoplasma. In cytoplasma there is a lysosome is also present. So lysosome and endosome fuse each other and form the phagolysosome. In this vesicle in phagolysosome there is a proteolytic enzymes are there. This proteolytic enzyme break down the peptide, a larger antigen to into small peptide like a 13 to 18 amino acid peptide. In this phagolysosome there is a highly acidic environment is created, so this enzyme give the maximum efficiency to break down the peptide. So this is how proteolytic digestion is occur. Parallel MSC class 2 already synthesized into the endoplasmic reticulum that will bind with the invariant chain that is called as a chaperone. This invariant chain already bind with the MSC class 2 because premature protein or else cell protein could not bind with this MSC class 2 complex, and that during the antigen is converted into peptide. This complex is bind into the vesicle, pack into the vesicle, and is head toward the Golgi body, right? So similarly, once this MSC class 2 molecule and this invariant chain it headed toward the headed toward the Golgi body, where in that complex it will this protein invariant protein is degraded by the enzyme. It leave only the small the clip for binding of the antigen peptide. So later on, this in the Golgi body there is a phagolysosome as well as this compartment cliff with the MSC class 2 molecule fuse is each the other. Cliff will release and by the HLA-DM, and instead of that binding the foreign peptide. So here this is a compartment in which MSC class 2 molecule, in between there is a foreign peptide that is 13 to 18 amino acid is bind on that vesicle. So now this Golgi body transport this vesicle with the MSC class 2 with the peptide on the surface of the professional antigen presenting cell via this vesicle move via the cytoskeleton. Now this cell is ready for the recognize the foreign antigen or foreign peptide by the T helper cell having CD4 positive receptor. Once it binding is happen, this T helper cell will release various kind of cytokines like interleukin 2, interleukin 4, 5, as well as interferon gamma. According to that, this give the action like it produce B cell produce more plasma cell, memory cell, as well as it plasma cell secrete the antibody. Similarly, it also release it activate more macrophage for enhance the phagocytosis process, and it also activate T cytotoxic cell. So This is the exocytosis pathway, how it run. This is a summary of the exocytosis process, in which exogenous pathway, in which you can see here, this is extracellular antigen and that enter into the cell via endocytosis for process. It form the phagolysosome parallel MHC class II molecule are already synthesized in endoplasmic reticulum with the invariant chain. This compartment is moved towards the Golgi body. In the Golgi body, there is a phagolysosome breakdown peptide, and this invariant compartment MSC class 2 compartment having a MSC class 2 and with the ear clip is replaced with the degraded invariant chain, and in this compartment cliff will release, and instead of that there is a binding of the peptide chain by help of HLA-DM. Later on, this vesicle move toward the cell surface. It is recognized by T helper cell. So this is the exogenous pathway. This is the MCQ regarding the exogenous pathway. You have to give me answer into the comment box. Thank you very much for listening this episode.